Okay, so I'm very glad to introduce our first speaker, Gideon Schreiber from the Department of Biomolecular Sciences at the Weizmann Institute of Science. And Gideon research focuses on the physiochemical properties of protein-protein interactions. And today he will tell us his recent and exciting results about the SARS-CoV-2 Omicron prediction and the antiviral drug design that enabled RBD in vitro evolution. And Gideon will first tell us 2 minutes about Instruct, and then he will start his talk. So Gideon, please.
Thank you, Michal. So Instruct-ERIC, I'm involved in Instruct for the last 15 years now. Michal Sharon is Israel representative. So until last year, I was with Joel Sussman. What is Instruct-ERIC? Instruct-ERIC is a pan-European infrastructure for structural biology and related. And as one can see here, it's related to research visits, training, R&D funding, and internship. You're welcome to apply. You have here the addresses, and you can get funds and opportunities for your research here. You can see it's really pan-European. There are 15 member countries right now in Instruct-ERIC, and they're spread all over Europe. And once you apply, you can get access to the fantastic facilities in these different countries, especially in the hubs, and the hubs are here with the red points. So please do and take advantage of what Instruct-ERIC can offer for you. Now I will go to my talk, and my talk is basically about SARS-CoV-2, Omicron prediction and antiviral drug design, and enabled by RBD in vitro evolution. I'm from the Department of Biomolecular Sciences at the Weizmann Institute. So I think that all of you already know about the spike protein. So this is a spike trimer, and it's binding the ACE2 protein that is on the cell surface. Upon binding to it, the virus is, the spike is cleaved, and then the virus RNA enters, and one gets infection. So what we decided that we want to make a drug in order to inhibit the viral entry. And this we decided in about March 2020, so it's now 2 years ago. Our idea was to take the structure, the protein of the virus, so this receptor binding domain of the spike protein, which you can see here also in yellow, and optimize it for binding to the ACE2. Now, this is ACE2. ACE2 is important for cellular functions. It's cutting a peptide. This is active site, so the binding of the virus is not at the site of the enzymatic activity, and we want to avoid disturbing this enzymatic activity, and I will show it to you later. So in the first step, what we did, we stabilized this receptor binding domain here. Then we optimized the inhibition of the receptor binding domain so that the virus cannot enter now. Now, this is an important point because the entry point, the ACE2, is something that is not mutating while the virus is mutating. So if we do the binding of our inhibitor to the entry point, to the cellular protein, then even though that the virus is mutating, it should not affect the efficiency of our inhibition. Now, our idea was to administer it through inhalation. Inhalation of proteins is less common than, or it's very uncommon, I would say. But from reading literature, it seems to us that this is something possible. So we worked together with Inon Rudik from chemistry at the Weizmann Institute to optimize inhalation techniques. And lastly, we did animal experiments, and I will tell you the story and how this relates to the Omicron. So first of all, if we speak about how do we optimize both the stability and the binding, then we used yeast display. Now, we used a methodology of yeast displays that we enhanced. So it's a much more efficient yeast display methodology. I will not really talk about it, but it's published. You can see here the reference for it. There are 2 references where these methodologies are published, and I welcome you. We put all the plasmids for it public domain, so everybody can use it and take advantage. So our first round here of selection was to increase the expression on the surface of the yeast. Now, when you increase the expression, very often you will stabilize the protein. And indeed, what we found is that the optimized expressed RBD on the surface here of the yeast was a mutation that is stabilizing by about 8 degrees the RBD, and it was a very stable one. Next, we did 3 rounds of yeast display selection. And what we did three selections in each round, and then we took not single solutions, but we took all the solutions that came out, so the whole population, and did again random mutagenesis on it. And this way, we always took the best binders. And lastly, what we did is a method that we also devised, which is called pre-equilibrium selection, where we take the selected clones that come out the first, so very rapidly. And this is in order to increase specifically the association rate constant, and not only to make the dissociation slower. So we wanted to increase association and dissociation. At the end, after all these rounds, we came up with 9 mutations, and you can see them here. And with a selection, so this is a stability selection, this here stabilized the protein. And then with each round of selection, we got additional amino acids that stabilize themselves. I think what you can already see that many of those that stabilize themselves during the in vitro selections that we did in the test tube, we can actually find also in lineages that we already know. So if you look at the alpha lineage here, then which it was common, came from Britain, but it was the most common one in summer 2020, we can see that indeed the N2Y mutation was a major mutation in the alpha. And then if you look at the beta and gamma of the Brazilian and South African, then we can see here that they came position 484 together with the 501 also stabilize themselves. And this is what we found also in other cases. So basically what we see here that we mimic previous selections of the virus, that the virus did it in population, but we did it in vitro. Another one of these mutants is the 477-N. Again, this is a mutation that was found before in a number of popular strains, as you can see here. So this is, so basically it looks at our in vitro evolution mimicked what the virus did in reality. Now we got also some other mutations here, especially remember this 498Q2R mutation, which we found as well. So this is affinity, and this is the starting affinity of the RBD to the ACE2, and you can see it's about 17 nanomolar in using SPR, and this is the affinities that we got, and we got about 16 picomolar. In other words, we got a 1,000-fold increase using yeast display in a relatively very short time of the affinity. Moreover, because we did this different rounds of selection, it also, we got a much faster on rate that you saw, it increased about 10-fold. So it's not only that the off rate got much lower, as you can see, but also the on rate got much faster. The structure of this was solved using cryo-electron microscopy within a few days by a fantastic work of Nadav Elad and Orlidim from our research facilities here of the electron microscopy and of the protein production facility here that we have. This is basically the center that this is the Instruct Center here. And what you can see also, one thing which is very clear, that you can see here that the positive charge of this optimized inhibitor, which we called RBD62, so now you will see always RBD62, got much more positively charged from what it started, so we get much better charge complementarity, which is a reason for the faster on rates. If we look more carefully at the mutations that we selected, then we can see that we have both epistatic and non-epistatic mutations. So the 484, together with the 501, which is a hallmark of the beta and the gamma variants, these two mutations are not epistatic to one another, and the interaction energy between these two, when you do a double cycle, is close to 0. On the other hand, if you look at the mutation 498 to R and 501 to Y, we see a large interaction energy. And the reason is that the 501 that you can see here, and this is from the structure, the 501 is really kind of stabilizing the 498R in its position so that now it can do salt bridges with an aspartic acid 36 and with Q42. So clearly, this kind of mutual stabilization of these two residues is very important in order to get higher binding affinities. On the other hand, you can see that the 484 and the 501 are far away from one another, so that they are not epistatic to one another. The next, we checked whether our inhibitor really works to inhibit the COVID, and we checked it against a number of different strains. So we checked it against the wild type, against the alpha and the beta and the gamma variants here. And what you can see here, that the IC50 and IC90 was hugely decreased. So you can see here, for example, from 23 nanomolar for if you use a wild type RBD to about 90 picomolar when we use the RBD 62. So we get few hundredfold increase in the binding affinity, and this is independent on the variant. So you can see here that the different variants are not affected. So basically we get kind of the same kind of inhibitory effect for the different variants. This was done in collaboration with Bjorn and Jean from Institut Pasteur. Of course, we wanted to check here that we don't inhibit the angiotensin activity of the ACE2, which is very important for blood pressure maintenance and other things. And what we see here, that the inhibition here, this here is an in vitro assay to determine the catalysis. So this is time, and this is a fluorescent signal. When you get higher fluorescent signal, then basically you follow the cleavage here of the peptide. And you can see this is done in vitro, and this here on cell lines. And you should compare the black line to the magenta one. And you can see that basically there's no difference, both if you do it on the cell surface and in vitro. Now, the fact that there's no difference on the cell surface basically means that also binding the RBD62 is not affecting it's not causing endocytosis. So it's not only that RBD is not affecting the enzymatic activity, it also does not induce endocytosis. So basically we get normal enzymatic activity of the ACE2 here, which is very important. Lastly, and this is collaboration with Daniel Duet and Matt from the NIH, we did non-human primate experiment. So basically we gave here 5 days the drug by inhalation, and we followed the amount of virus. Now we are doing many more tests in order to see what happens to disease burden and not only to the number of viral particles. And this here's copy number in logs. So if it's, let's say, this would be 1 million and this would be 10,000. And what you can see here, that at day 2, we get a huge decrease in the amount of viral particle. And this here is done close to the lung, so the measurements are deep inside where you really want to avoid the virus, because this is where most severe disease comes from. And you can see that also after 5 days we stop treatment, the virus count is much lower also days after we stop the virus. And at day 9, for example, here you can see that in half of the animals there's no viral load at all anymore seen at day 9. So in other words, it seems that the strategy and that we used here by making a tight inhibitor and giving it through inhalation is working. And now we are looking for companies to bring it further on for clinical trials. Now, as I already have shown you, there is something related between the Omicron and our in vitro evolution. And between, in general, I would say virus evolution and between virus evolution and the in vitro evolution. And what we can see here is the changes, the mutations of the different main variants, and this here is the ACE2 binding site. So you can see here this ACE2, and this is alpha mutant, the 501, the delta mutant with the 452 mutation and the 478, which are the major delta, and this is a beta mutant. Again, it's all close to the binding site, and now we see the Omicron, and in Omicron, we got a huge amount of mutations in the area of the binding sites, over 30 mutations plus deletions and so on, and actually the Omicron keeps on evolving, as you all know from the news. And now if we look at our in vitro evolution, we can see that this 498, which did not come up before, now in Omicron, and we were looking for it, right? We actually said, okay, this seems to be really important, this 498 for viral evolution, because it's increasing the binding affinity substantially, but we couldn't find it in previous variants. And then in November, we saw the sequences of the Omicron, and we said, wow, you know, that's what we predicted, that we'll get both the 501 and 598 together. And in the Omicron, we also have the 484 and also the 477. So all these mutations that we predicted could be problematic from our in vitro evolution came up with the Omicron. So now we had a huge collaboration with many groups, and this was led by Gideon and David from Oxford, and we participated in it together with many other people from around the world. So what we did, our part in this collaboration was really the work on how the binding interface changed in the Omicron and how this relates to binding affinity. And you can see here also that many of the residues and all are all marked here, and the interface are changed in the Omicron relative to what happened before. Also, the Omicron had a much higher positive charge, like our RBD62, and this is the negative charge. So you see increase in positive charge, so better charge complementarity here. So we analyze the binding interface, and if you analyze the binding interface, what you can see here, that the 498 by itself, the mutation is actually reducing the binding affinity, so binding is less tight. But once you combine the 498 together with the 501, you get a huge boost of about 30-fold in the binding affinity. If you add additional mutation found in the Omicron that we also found in our cases, like here, this is kind of what we found in vitro evolution and in Omicron, we get an increase of almost 40-fold in binding affinity. Now, surprisingly, the Omicron is not increasing binding affinity, as you can see here. So what happens? Why isn't binding affinity increased if the mutations that we predicted increased by so much the binding affinity? And the reason for this is that there are other mutations in the Omicrons that actually reduce binding affinity. And this includes the 496S, and this includes 3 mutations here of serine to leucine, proline, and phenylalanine, which I will discuss in a moment, and additional mutations as well, like the 417 by itself, and so on. So what you can see, that what happens actually in the Omicron is that the improved binding affinity that is due to the mutations that increase the binding affinity within the interface allowed the interface to change, and so to put other mutations inside the interface and in other areas that will back reduce the binding affinity. So what is the importance of these other mutations? Why is all the mix so important here? And this can one see very nicely if one looks where are the main binding sites of the antibodies, neutralizing antibodies on the surface of the RBD. And one can see here, these are the potent antibodies, either of the or the therapeutic antibodies, and this is the binding site. And here, this is a hot area. And you see the mutations in the Omicron are basically located within the hot area, both the therapeutic antibody binding site and the most potent binding sites of the pandemic are basically in the area where there are mutations in the Omicron. In other words, the Omicron is using the increased binding affinity in order to put in new mutations that now will decrease the immunity of people who either got vaccinated or had the disease before, because now the huge number of the majority of the world population already was either infected or got the vaccine. So the Omicron, in order now to be so efficient, has to overcome this, and it did it by this kind of rechanging the binding site. And this here, you can see here how the neutralizing antibodies that were developed and the commercial ones, you see the black line for Omicron, and you see that they don't work anymore except for one. So this one is the only one, neutralizing antibody commercially available, that still works against Omicron, while all the others really lost their ability to work. And indeed, now for Omicron, the neutralizing antibodies are not used because it's kind of useless. And one also can see that the amount of antibodies from people who got the vaccination. So you see here, this is a vaccination. This here is kind of a higher number means more, okay? And you can see that after the second vaccination, one gets here with the Victoria strain, the initial one, a high number with time, it's kind of fading. And then with the booster, the third vaccination, one gets a big increase to a number of kind of 9,000, okay? With the Delta, we get the same picture. So we got a large decrease before the booster, and indeed the booster did a great job. When we look at Omicron, also the booster is doing a job, but you can see that this number is much lower than after the booster of the Delta and clearly the Victoria. And indeed, the vaccination is much less efficient against Omicron, but it's less efficient for people to get the disease, but it's still very efficient in avoiding severe disease. And at the end, what we want from a vaccine is to avoid severe disease. And indeed, people who got vaccinated, the chance of getting severe disease is kind of at least tenfold lower than people who are not vaccinated. So the difference is huge, and one can see that a majority of people who got severe disease are those that were not vaccinated. So vaccination is really important here, although it's not really affecting much the possibility of getting sick. So if I summarize it, neutralization titers of Omicron by sera from vaccinated and convalescent subject infected with early pandemic are substantially reduced. All the sera failed to neutralize. Titers against Omicron are boosted by third vaccine doses. Mutations in Omicron knockout or substantially reduced neutralization by most of the pattern antibodies under commercial development. Omicron spike structure as structural changes from early virus and uses mutations that confer tight binding to ACE2 to unleash evolution driven by immune escape. This leads to a large number of mutations in ACE2 binding site and rebalances receptor affinity to that of early pandemic viruses. Now, Omicron shows that drug development is an important option for the long run and should be pursued, because clearly one can imagine that other variants will come up in the future. And of course, this is something that a drug could solve if the drug would be good against all different variants. And of course, this work is not done by me. And the most important person, which I didn't show you a nice picture, sorry, for him is Yerke. Yerke is a Czech postdoc who is totally amazing. He was a driver in my lab. He did much of the work that I've shown you here. And yeah, and now he's looking for a faculty position in Czech Republic, and I'm sure that he will find also one. And I have to thank many others from my group who helped me in doing this, and Orly and Nadav, who solved the structure, Yunun Rodich and his lab for the inhalation and all the development of the inhalation processes. Of course, the University of Oxford for the fantastic collaboration on the Omicron, the NIH for the animal experiments, Institute Pasteur for cell experiments, our structural proteomic unit who helped in kind of making this come true in the protein production, and of course the funding agents. Without them it would not be possible. Thank you very much.
Thank you very much, Gideon. We already have several questions, so maybe if you can stop the share mode, we'll be able to see you. So I'll start with one of the questions.
Here's a picture. Here's a picture of Yuri without the mask. Sorry. Now you see the mask out there. It stops the share mode.
Thanks. So one of the question is, do you think this in vitro technique could efficiently used to predict new strains, which could be used to prepare the messenger RNA vaccine before the outbreak, to make some kind of messenger RNA mix of potential new strains?
No, this is an excellent question. And from the beginning, that's actually one of the things that we said, yes, we can predict, and we predicted even the Omicron, although we kind of were not sure, we said, okay, 498 is so important, but why don't we find it? And then came the Omicron. So yes, we predicted. Now we have a very large project in our lab to make much deeper mutagenesis into the RBD and to see all the possibilities that it could maintain kind of similar binding affinity by changing kind of the surface in order to see which of those mutations now could be the worst in immune evasion. Because what is clear from the Omicron, the future variants have to do immune evasion because the majority of the population got either sick or was vaccinated, or many got both sick and were vaccinated. So a virus, in order now to infect, really needs kind of to circumvent it, and this is something that we're working on. So yes, I think you can at least many of these mutations predict.
Okay, thank you, Gideon. Another question from, we have lots of questions. I don't know if we have time. From Jose Maria Carrazzo, she says, thank you. And she asks if Omicron is supposed to be very transmissible, but you are finding that it is not that good in terms of increasing AC2 affinity, but it is very good in invading antibodies. So the virus gets less into the cell, judged by ACE affinity, but it's not controlled by antibodies.
So I think what he wants, so the binding affinity of the Omicron is about the same like the original virus. So it's not that it's binding weaker. So apparently if it would bind weaker, then you would start to pay a price. Very often it's kind of, there's an optimal binding affinity, and apparently binding affinity of the original virus is kind of okay. So what it did, it kind of took advantage. What is more important, maybe to increase a little bit binding affinity or to avoid the immune system, probably avoiding the immune system, because the majority of people now have immunogenicity against the original virus. Probably this is much more important in order to get spread. So this is kind of, okay, viruses don't think, but this was a winning solution.
Another question is about the new strain of Omicron, BA2, that in addition to the Omicron mutations also bears the N501Y. Is it predicted to have increased?
Okay, I can answer it without it because it's clear. So the 501, so first of all, the Y501 mutation is already in the original Omicron. So I guess you speak about other mutations. Yes, the 2 variant of the P2 variant of the Omicron has a number of other mutations. Why they are important is not that clear. There are some talks that it may be more infective. I don't know how kind of solid this data is that it's really much more infective. And so I cannot say much more, but the basic architecture, all these mutations that change the binding site in the Omicron, I actually also found on the B2. So this didn't change. What changed were a few more mutations. One of them is related to, I talked about these 3 serine mutations and I didn't say much about it, but basically this is a site where you get lipid entry. And what it seems that the Omicron does not require anymore the step of cutting the spike, but it can enter as a whole virus relative in the higher part of the trachea and doesn't go so much into the lungs. And probably this is also why it's much more infective, but why the disease is milder. And there is a possibility that these additional mutations will relate to the same thing, that it does not go deeper into the lung, it stays higher up and so can be even a little bit more infective.
Okay, I'll ask you the last question, which was actually the first question that was in the questions. And then Gideon, I'll ask you to answer the rest of the questions by text. So this question is about the yeast display protein stabilization. It looks like a fantastic technique. Would you expect that this could be applied to transmembrane proteins as well?
I have no idea, because yeast display with transmembrane proteins, we never ever did. I don't know anything about transmembrane proteins, whether you can, how good it would be to do selection with transmembrane protein, because yeast selection works on showing a soluble protein on the surface. So I don't know whether you can do any transmembrane proteins on yeast selection, but that's kind of just, I have no idea. So I don't want him to say something about it.
So thank you, Gideon. Now you have a lot of work answering the remaining questions by text, and we'll move to our next talk. So our next speaker was supposed to be Sarel Fleischman, but he's now with Omicron at home recovering, and we are lucky to have with us Shiran Barber-Zucker, a postdoctoral student from Sarel's lab, and she will be telling us about AlphaFold and protein optimization. And as you can understand from the title, Sarel's lab and Shiran are focusing on computational biology. Thank you, Shiran.
Thank you, Michal. Thank you for the introduction. And thank you, everyone, for joining to the seminar today. So I'm Shiran. I'm a postdoc in Sarel's lab for about 2 years now. And I'll be talking today on behalf of him, as COVID successfully reached him just a few days ago. So our lab's main goal is to understand how function is encoded in proteins, and by such understanding, to develop tools that can optimize existing activities or even design completely new ones. And over the past 5 years, the methods that were developed in the lab have been proved to be very general, and we can now apply them, the same strategies to many kinds of protein, from binding proteins to vaccines or enzymes, and achieve dramatic improvements in different aspects. Today I'll start my talk by describing one of our main optimization approaches that depends the screening of only few constructs in order to find much improved variants. And in the second half of my talk, I'll discuss one of my projects and how the AlphaFold revolution actually generalized our approaches even further, and how the combination of these new generation structure predictors and our methods can enhance the utilization of very complex proteins. Okay. So first, I should start by saying that we all should understand that engineered proteins are already part of our lives, and they are critically utilized in many industries, from paper and pulp industry to the food industry and food tech, and of course in pharmaceutics. And with global warming, it is also very clear why the chemical industry is moving to enzyme-based processes, which are much more economical and environmental friendly. So we understand that proteins are great and that they are needed for many applications, but natural proteins are simply not good enough for biotechnological or biomedical applications. For instance, they cannot withstand high temperatures that are needed for industrial processes. They cannot be expressed efficiently in relevant organisms. Their catalytic rates are poor. They are sensitive to environmental conditions, such as the use of different solvents, and they often don't have the required specificities. And sometimes, and quite often, there are also some trade-offs between such traits where, let's say, an enzyme variant is highly thermostable, but it is less active compared to a different enzyme that is less stable but more active. And then we have to make some difficult decisions on which variant we want to use. And the main strategy today for optimizing proteins relies on evolutionary processes for improving a certain trait. We start with a native protein, and by accelerated evolution in the lab, the protein acquires beneficial mutations, usually one at a time. And you can think of it as a series of steps within the fitness landscape that samples the surface of the landscape, and the screening or the selection actually paves the route and paves the way towards the fitness peaks. But while directed evolution can reach the peak and to the maximum activity levels, it can also be trapped or stuck at local fitness maxima, where it cannot cross some fitness values and access other fitness peaks. This process is quite laborious and demands the screening of usually thousands of clones for the optimization of a single protein for a specific selected trait, and therefore they cannot be effectively applied on many starting points. So the evolutionary methods are not only laborious and time-consuming, but the fact that we rely on chance to generate new enzymes, binders, or life-saving drugs reveals some critical gaps in our understanding of how function is encoded in protein sequences. And what Richard Feynman have described generally brings us to one of the fundamental issues in protein biochemistry. As long as we don't know how to create the desired function, we don't really understand how the function is encoded in proteins. And this is maybe the defining question for the field of protein design. Can we close these gaps in our understanding and come up with a general strategy to design new or improved activities? So this slide summarizes the past decade of the lab's work, trying to bridge up these gaps. In atomistic protein design, we define each and every atom in the protein structure and compute the system energy. And by mutating the protein or changing a specific conformation, we can optimize the system, aiming to find a low energy solution. But while this atomistic strategy works great for small and simple proteins, the lab and others have found that in real-world problems or real-world proteins such as enzymes and antibodies, the atomistic design strategy on its own, it simply fails. And as most natural proteins are too large and complex for atomistic calculations to design accurately, the lab have developed a strategy, which we call evolutionary guided atomistic design, that uses information that is encoded in sequence alignments and in structures in order to optimize atomistic design of more complex systems. And in my talk, I will show you how we can utilize the this new generation deep learning structure predictors in order to solve important problems in protein engineering that were thought to be intractable by computational methods before. So one of the most fundamental problems in protein science and engineering is protein stability, as many natural proteins are only marginally stable. And as many enzymes engineers know, mutations that enhance activity often negatively impact the stability and foldability, and vice versa. So we wanted to find a way to stabilize proteins, but without affecting their activity. And we didn't want to have a specific strategy that can be applied only to a certain type of proteins, but we wanted to develop a general design strategy. And this is when Adi, which is an amazing scientist and was a PhD student in the lab, has developed PROS. So PROS, or the Protein Repair One-Stop Shop, is a general algorithm that uses phylogenetic information and Rosetta atomistic calculations in order to optimize the native state energy of the protein. PROS apply mutation only outside of the active or the binding sites in order to maintain the native function of the protein. But it therefore means that almost all the protein is allowed to be designed. And to narrow down the sequence space in every designable position outside of the active site, we allow only substitutions to amino acids that are first found in homologous proteins in this exact position. This means that we allow only substitutions that are conserved in evolution, as we expect that during evolution, the selection pressure lead to tolerated substitutions. And second, out of these substitutions, we allow only substitutions that by themselves, as block mutations, improve the native state energy of the protein. Then in PROS, we do a combinatorial design and suggest about 9 low energy designs with different mutational loads. In some cases, we are suggesting more than 10% of the protein sequence to be mutated. And I will show you only one short example where we applied PROS to acetylcholinesterase. This is a very essential enzyme that have a role in neuromuscular junctions and is a target for nerve agents, but it is also a very big and challenging enzyme for production. Our best PROS design possesses 51 stabilizing mutations. This is a huge number of mutations. At least this was a huge number for us for the time that this study was applied. And as many protein engineers knows quite well, introducing only few mutations usually leads to loss of function. But all of our designs were fully functional, and they were much more thermostable compared to the native protein, and showed in some cases 2,000-fold improvement in the protein, in the bacterial expression levels, actually enabling the expression of this very complex enzyme in bacteria, which was not possible before. So after using PROS in the lab for the successful design of different kinds of proteins, Adi made it fully automatic and applied it also as a web server that is accessible to any academic user and does not require any expertise in protein design. And ever since, PROS was used by hundreds of users around the world. And there are already dozens of papers that have used this web server and other web servers from our lab in order to optimize proteins for diverse applications in different fields. But protein design methods, and PROS is no different in that sense, require highly accurate atomistic models in order for the designs to be reliable and for their success. And this means that the scope of proteins that we were able to design was very limited to those proteins that their structure was determined. However, challenging proteins that are impossible to express in heterologous hosts usually don't have a crystal structure, as they cannot be produced in sufficient amounts, or that they are not stable enough for structural determination methods. And this is like a chicken and an egg problem, because we would benefit most from using design to stabilizing these proteins, for example, in order to enable the structural determination. But this design is enabled only if an accurate structure exists. The last year structure prediction revolution that was led by DeepMind team and David Becker's lab encouraged us to test whether the new deep learning-based predictors produce models that are accurate and reliable enough for design. This would mean that if we are freed from the need in experimental determination of structures, then our methods, and especially PROS, could be utilized on any protein or almost any protein you have in mind, and can enable the experimental exploration of very complex enzymes that could not be that simply could not be studied before in the lab. So in recent years, our lab became more and more interested in finding solutions to environmental issues. And in this case, this exact same chicken and egg problem arises because many of the enzymes that are relevant for such applications are very hard to combinationally express, and therefore could not be optimized by our methods. And furthermore, they are also far from being as thermostable as they are needed and having good activity rates that are needed for industrial applications. And one such problem that we chose to tackle is lignocellulose degradation. Wood and plant biomass are the most effective carbon resources that we have on Earth and that can be used for biofuel production. Plant cell walls are comprised of lignin, cellulose, and hemicellulose. Lignin is a polymer, a complex polymer that is made by cross-linking aromatic precursors. And this is the component that actually gives cellulose the rigidity. But in order to utilize the cellulose for releasing the fermentable sugars that can be then be used for biofuel production, pre-treatment is required in order to loosen the lignocellulitic complex and to make the polysaccharides accessible to hydrolytic enzymes or to chemicals. But because lignin wraps up cellulose and hemicellulose, it obstructs the accessibility of such chemicals and enzymes, and therefore impedes their conversion into biofuel. But lignin is a very complex polymer, and its chemical degradation requires very harsh conditions, and therefore it is far from being sustainable. Luckily for us, Mother Nature have emerged lignin-degrading fungi about 300 million years ago, and therefore the option of biodegradation for environmental friendly utilization of wood biomass is highly relevant. So the only fungal enzyme family that degrade lignin by its direct oxidations is versatile peroxidases, or VPs. They have very high redox potential, reaching about 1.4 volts. And therefore, they are also of special interest for their utilization for pollutants or even plastic degradation. However, although hundreds of VP sequences are available, there is only one VP that the structure was determined and that was subjected to intense biochemical characterization and engineering campaigns. And they are very hard, if not impossible, to be produced heterologously in functional way. And this is not surprising when you are considering the structural complexity. About half of the VP structure is composed of loops. Unlike regular enzymes, they don't have one active site, but they have 3 distinct active sites: an active tryptophan, a heme channel, and a manganese oxidation site. They have 2 structural calcium ions, and they have multiple glycosylation sites and disulfan bonds as well. And the fact that we have only one crystal structure means that most of our knowledge of this family arises from the study of a single enzyme, and the natural functional diversity in this enzyme was simply could not be studied. And this is also true for many complex protein families, where most of the data comes from a representative protein that its structure was solved, which again means that the functional versatility remains a mystery. And with that in mind, we chose to examine whether the new structure predictors can be used as templates for post-design, enabling the exploration and the optimization of these challenging enzymes such as VPs. So in this study, we chose diverse VP sequences that span the whole phylogenetic tree. We then modeled them using TR-Rosetta. TR-Rosetta is a deep learning-based structure prediction algorithm that was released in the end of 2020, so about a year ago. And it showed dramatic improvement in structural prediction compared to previous methods. Of course, that ever since AlphaFold 2 and RosettaFold became available, and they are much more accurate predictors, and which therefore it makes TR-Rosetta like an ancient history. But at the time that we started this study, TR-Rosetta produced very good models, and it was the only one who was available. So we used TR-Rosetta, and then we cross-designed each of these models or this protein to stabilize them and hopefully to enable their functional expression in yeast. And here we chose to test 3 designs for each sequence, with about 3 to 12% of the sequences being mutated. And you have to bear in mind that the ability to design 30 or 40 mutations based on a model of a very challenging enzyme and observing gain of function demands super high accuracy in both of the modeling strategy and of the design method. And indeed, initial scanning revealed that for 4 out of the 12 sequences that we started with, we obtained functional expression. This is the activity axis. And you can see here that the most successful designs, and you can see that their activity depends on the mutational load. The wild-type protein here in gray exhibit poor functional expression, if at all, while for all the sequences, the designs with the highest mutational load, here in green, are the most active designs. And these are designs with approximately 37 to 45 mutations in each, which therefore reflect for the high accuracy of the models that we used. This is also reflected by the fact that we were able to introduce stabilizing, but sometimes radical mutations, for example, to improve core packing. And you can see the design models in purple and the wild-type in gray. PORS also improved loop rigidity and created new hydrogen bond networks between several proximal mutations. These are the same kind of mutations that we observed when we applied PORS on crystal structures. But again, the ability to insert 3 mutations and create a network of interaction wouldn't be possible if the accuracy of the models wouldn't be sufficient. So the next step was to characterize the best VPs for their stability and activity profiles. And we tested out our best 3 VPs and compared them to R4 and 2-1B. These are 2 VPs originating from the same protein coming from an homologous fungi that were previously engineered by directed evolution for high secretion levels, R4, or for thermal stability, 2-1B. And this was done by screening 5,000 to 9,000 clones. And I'm not going to get into too much details. I will just say that all of our VPs that we designed have much improved expression levels compared to the best expressing VP that was evolved before. And all of them are more thermostable compared to this variant. One of them, in some manner, is even more stable than the variant that was evolved to withstand high temperatures. And furthermore, all VPs exhibit high stability and varied pHs from acidic to basic pHs, which is highly relevant for their utilization for different biotechnological applications. When looking at the activity profiles of these enzymes, the picture is a bit more complicated. I don't want to get into too much details here. I would just say that we tested their activity against 5 different substrates each and every one of them have a unique chemical properties, but they are also targeting different catalytic sites, which means that we examined the activity of all the 3 sites. And the different VPs show distinct activity profiles, with each and every one of them exhibiting high catalytic rates and high affinity to different substrates. And most importantly, there is no one VP that is best against all substrates or a desired pH, but each and every one of them is good for a specific functionality. And the design of VPs was kind of a proof of concept for us in that sense that there are more fungal enzymes that participate in lignin degradation. And by using this platform, we aim to explore the functional diversity between enzymes belonging to each family. And as the functional profiles of VP suggest, designing a enzymatic cocktail that is composed of enzyme repertoire might degrade the complex structure of lignin much more efficiently than the use of a single enzyme from each family. Last but not least, this approach also enabled us to study the structure-function relationship of the proteins in study. Personally, this was highly interesting for me. I'm a former crystallographer, and I'm used to use structural data in order to understand how protein functions. But until now, there was only one crystal structure of a VP that was available, and this VP is the only one who was exhaustively characterized, making the structure function studies of these enzymes quite speculative. However, again, the fact that we were able to introduce 40 mutations to the models means that they are highly accurate, and therefore we can use them to try and explain the functional versatility that we observe. And indeed, the most different VPs, both in sequence and in function, we call it VP8, show some variations in structural elements like this extra loop and some differences in other loops conformations around the protein. And when we look at each and every one of the active sites, the manganese sites, the tophanol sites, and the hemaxis channel, we can see and spot some variations that might cause for the differences in function. And because in POST we are not designing the active sites, these are naturally differences that are originating from the native sequence. For example, variations in the heme channel that lead to distinct electrostatics and shape of a specific enzyme might be the reason for the high affinity but low cooperativity rate that we observed for this enzyme to a specific hydrophobic substrate, and compared to the others, to the other VPs. So before I finish, I just want to add that the lab has developed other methods for protein optimization, and you are more than welcome to visit our website and access the relevant servers. I just want to mention one of them called FuncLib. FuncLib is similar to POS in terms of the rules that we use in order to select mutations, but focuses on the design of the active site in order to improve the enzyme activity or change its specificity. FuncLib is also applied as a web server and can be and was used by academic users without design expertise for improving protein function dramatically. And together, if you view these methods as a whole, you can see that they present a completely computational test to protein optimization, design of the active site using FuncLib in order to improve the activity, and outside of the active site with POS in order to improve the stability. There is very little previous knowledge that is needed of the protein activity in order to use these methods, and they are much more effective than iterative in vitro evolution studies. And of course, these methods are completely automated, and now with the accessibility of accurate models of almost any protein you have in mind, and we already integrated the use of AlphaFold models into the POST web server. These methods can be used to design effectively many homologs for bioprospecting, for example, design enzymatic cocktails as we envision for immune degradation, and to design pathways for synthetic biology projects. And with that, I would like to thank you all, and if anyone has questions, I'll be happy to take them.
Thank you very much, Shiran, for this interesting talk. We already have one question from Lorenzo Rossi. He asked, does PROS algorithm work well also for membrane proteins?
Okay, so we have developed a special PROS version for membrane protein. I'm not part of this project, but it's not accessible yet to everyone, but you're more than welcome to contact us, and it does work. We know that it works for membrane proteins as well.
Okay, I also noticed that one of the participants, I don't know whom, raised his hand. So if you would like to unmute yourself and ask a question.
Hello, I was wondering whether it could help when you've got, for example, insertion domains that are flexible. Can it help you to suggest how to cut it out, or what's the best short region you could put instead of, let's say, a 200 amino acid long disordered insertion?
Protein design can be used for these applications. Specifically, the methods that we developed in the lab are not touching these problems, but generally protein design can be helpful to predict or to understand what would be the best solutions for such problems or substitutions.
Thank you.
Shiran, we also have a question from Salvador Guardiola. I'm sorry if I'm not pronouncing it right. Great talk. Maybe I missed this, but how are residues for point mutations initially selected? Is there rationale or are they stochastically tested?
So in PROS, we actually enabled design of all of the protein, except of regions that relates to the activity, meaning if we have the active sites or binding sites or oligomerization sites, we don't touch this region in order to not defect the native functionality. But other than that, all the other positions are, can be designed.
Okay, we have another question from Rodolfo Razia. He's asking, I was wondering about the manganese 2 site. How well is it modeled and how reliable is it? Considerate AlphaFold does not take metal sites into consideration.
Yes, so we were made to see how well all the active sites in VPs, how well the conformation was. So the manganese site, even the heme site, which is heme is a big molecule that's supposed to be inside of this protein, and we don't model it using these structure predictors. All of them were well aligned to the known crystal structure that we know, and they all made a lot of sense when we insert the manganese site after modeling. So it handles it very well.
Okay, so we have an interesting question from Mike Walker. He's saying, since you just spoke after our coronavirus talk, I wonder whether you gave any insights into how Omicron variant with so many amino acid changes emerged.
So I'm really not involved in any of the coronavirus projects in the lab. I have no idea. Maybe Israel will have a better answer if he was looking at it. So you can address him this question directly.
Okay, so if Israel, if you're here, you're welcome to stand up. And I wanted to ask you, so AlphaFold and the Rosetta fold still have problems with certain kinds of proteins, like we just heard, like that, for example, metal binding. So where is the limit of your current method in combining this response?
So of course, in this stage, we would suggest to do it with cautious. You need to look at the structures and the models that AlphaFold produced before you are designing them to try to understand if the model looks okay and if it fits what you expect it to look like. AlphaFold specifically gives you the data about the accuracy of different regions in the proteins. And we don't allow designs in these regions, in PROS or in any other algorithm that we have. So we take this into consideration. And of course, time will tell.
Okay, and Sarel is here with us, so maybe he can answer the question we had before. And with this, we will end. Sarel?
Sure. Sorry, so I'm not 100% sure how to answer this. It's a very interesting question. What we should understand, and especially I think in view of Gideon's talk in the first part of this seminar, is that the number of mutations that the virus can undergo is just huge, and there's a lot of there are questions related also to chance. Which mutational path the virus, let's say, chose sometime in the beginning of the pandemic can dramatically affect the choices it has right now. So I think a priori predicting what mutations, what mutational paths would be taken is quite difficult. We may be able, as Gideon showed, to predict maybe that a few of the mutations that are adaptive, but I think in general it would be very difficult. There's probably a huge number of combinations and mutations on the surface of the spike that could be tolerated and even beneficial. And which ones are actually taken in the field is not easy to predict.
Okay, thank you. Thank you, Shiran. Thank you, Sarel, for the question. And thank you, Gideon, for the first exciting talk. Thank you all.